So this slide goes through some of the key terms that are needed for this part of the topic. At the top of the slide, you can see three keywords in capital letters: gametes, zygote, and embryo. And I'm going to talk through these in the diagram and explain how they fit together. So we'll start off with the gametes. The gametes are the sex cells, so the sperm and the egg in humans, and they contain 23 chromosomes each. So that is the haploid number, half the amount you'd expect in the body cells. At fertilization, when the sperm fertilizes the egg, the genetic material, the 23 chromosomes from each, join together to now form 46 chromosomes, or 23 pairs, and the fertilized egg is now called a zygote. The zygote contains all of the genes for the individual, and the zygote will now start a process of cell division. This cell division is mitosis, and this is where all of the genes are copied and they are identical cells being created. This cell division continues for about 2 weeks, up to the point where we form something called the embryo. And at the 8 cell stage, which is highlighted here, all of these cells are unspecialized. This means that they can develop into any other type of cell during differentiation. These are the embryonic stem cells that we hear about when we are thinking about this topic. If you remember back to the lesson on differentiation, all of these cells have got all of the genes turned on at this stage. None of them are switched off. This means that those cells can change into any other type of cell. Following on from this stage, these cells will continue to divide by mitosis and become specialized, and during this process some of those genes will be turned off so that those cells will have the information that they need to create the proteins that they need to function. So whether it be a nerve cell or a muscle cell or a skin cell,